So now I want to show you a couple of experiments that we did to evaluate these classifiers. So the first one is called the 5-fold cross-validation. So we take one data set, we split it into 5 folds, 5 groups, and each time you use 4 for training, 1 for testing, and then you shuffle them. This is a very unrealistic experiment, right? So usually you want your reference that you use for training to be totally independent from the testing. We did this to be able to compare the classifiers in a fair manner, right? That we don't consider also cross dataset differences and so on. We judge the performance based on 2 metrics. So the first one is called F1 score. So it's a bit of a summary of the precision and the recall. So higher is better, basically. So if you're close to 1 or 100%, that's perfect. If you're at 0, that's totally wrong. And we also look at the percentage of unlabeled cells. So for these classifiers that reject cells, they give you unknown cells. It's important to look at this percentage because very naively, if you reject everything, if you say everything is unknown, then your F1 score will be 1. You'll be doing perfect if you say, I don't know anything. So it's important for those to look at both values. Okay, I'm going to show you a few experiments. So these plots all look the same. Huh? So these are all the classifiers that we compared. On the columns here, you have the different data sets. So the first group of data sets are actually very easy data. So these are all the 5 pancreatic data sets. What you see here is that all classifiers actually classify this data easily, because if you have looked at pancreatic data, I guess maybe you have looked at it in the course as well, they're really easy to look at. You have relatively few cell types, and they are very well distinct because they also express these hormones like so you have the beta cells making insulin, you have the alpha cells making glucagon, and they're really very far away from each other. Cell bench is data set, to be honest, that was suggested by a reviewer. It's a cell line data set that's, again, 3 classes, very, very distinct. So if you make a mistake here, I think it's just bad. At the bottom here, you have classifiers where I have gray. So these are classifiers that we actually could not test on these data sets because they require prior knowledge. So they require that you give a set of markers for every cell type. And it's not that we don't know the markers of pancreatic data, but it's just to show that, you know, if you require these markers, it is a bit of a limitation. Most of these classifiers provide markers for a couple of data sets, mostly PBMC data, blood data. But if you require that I give you markers for pancreas, I can find the markers. That's easy. But if I look at brain data, for instance, markers for cell types are notoriously difficult. So this is a bit of a limitation for methods that require these markers. As I said, you should look at the percentage of unlabeled cells because it gives you an indication. So here, for instance, SC Predict is doing quite okay on this data set. They have an 0.95 F1, but they are rejecting 11% of the cells. So I would take another classifier that here, for instance, the off-the-shelf SVM is 0.99, and it's not rejecting anything. So this is certainly way better than this one. Okay. The next data set we looked at is data from the Allen mouse brain. So the reason we included this data is that you have one data set that's annotated at 3 different levels. So at the very highest level, they only differentiate between inhibitory neurons, excitatory neurons, and non-neuronal cells. So that's, again, a very easy task. And what you see here is that all classifiers do, sorry, all classifiers do well on this problem. On the second level, you have 16 classes, so it becomes a little bit more challenging, but still you see most classifiers are doing quite okay, not KNN, by the way. And at the deepest level, they have 92 classes. So this is, again, a very challenging problem. You can imagine that some of these classes are actually very, very small. There are only very few examples to learn from. And there you see that most classifiers have a lower performance. Again, look at both patterns because SVM rejection here. So this is just a typical SVM classifier with a rejection option. Has a very good performance, but it's rejecting close to 20% of the cells. So you will get 1/5 of your data without a label. So this is a bit deceiving. But in general, as you go to more deeper annotations, most classifiers have a lower performance. Yeah? But they suffer in a different way. So if you look here, SCVI, this is based on a variational autoencoder, suffers really quite a bit. And this is not because SVI is bad at all. This is just because at this level, there are a lot of very small populations. So this variational autoencoder cannot learn from this data at all. Okay, I did talk about this trade-off between the rejection and the performance, so I'll go over that. Okay, now let's move to PBMC data. We did include two data sets, both from 10x. So the first one over here is the sorted data set. So this is fact sorted and then single cell RNA-seq. And this one is just single cell RNA-seq and then labeled naturally. So here we could test these classifiers because they provide markers for PBMC. One thing to keep in mind, I have a lot of these asterisks over here, and that's because I can only test the classifier on classes for which they give markers. So SCINA only gives markers for three cell types in PBMC. It is doing perfect, but only in classifying 3 populations, while the dataset has 11 labels. Yeah. The others are also doing quite okay, but still they cannot classify the 11 populations because they only gave markers for less than this. Yeah. We also experimented a little bit with defining these markers ourselves using differential expression. That's what you see here, right? That I assume now that I don't have these markers, so let's just do differential expression, define the markers, and then give them to the classifier. You still see that it doesn't do very well over here in comparison to classifiers that do not actually care about these markers. So at least based on PBMC data, our argument is that don't use these prior knowledge classifiers, don't require markers, just learn from the data what's in it. It might not align perfectly with your biological knowledge that T cells should be positive for CD3, but it doesn't matter, right? Okay. What I haven't said is that we ranked the classifier over here based on their performance across all these data sets, right? And what you see here is that at the very top, the first 3 classifiers are SVM classifiers, and that's why I also tried to explain that a little bit. So SVM seems to be doing quite well here. What you also see is that SVM rejection and SVM, these are just scikit-learn classifiers that has nothing to do with single cell. They are doing remarkably well compared to other classifiers. So again, back to my point about generalization, the more you tweak the classifier, the more complex you make the classifier, the less well it will generalize to a large set of data sets. Yeah. Okay, I hope this is clear. Do you have any questions on this part?
Maybe a bit, sorry. Somebody. Yeah, I was just wondering, like, how in the end then do you make up your mind about which classifier to pick?
We'll get to that. We'll get to that. Remember, this is a very biased experiment that I'm showing you. So far, don't make choices just yet. This is a very biased experiment. Here we train and classify on the same data set. You will never do this, right? This is never your goal. So just wait a little bit. There was another question or another remark.
I was just wondering if different methods are like better for different tissues or.
Yeah, again, so this is still very biased. I'm not sure if we learned anything about this. What we learned is that some tissues are actually more complex than others. So that's where I was going. So let's talk about it here. So we tried to look a little bit into this. So how complex are specific tissues and how easy is it? So there are 2 factors. So one is how many cell populations. So if you're trying to classify 100 cell types, that's certainly more difficult than 5. But it's also, if these 100 are very, very distinct, that's easier than if the 5 are overlapping. Yeah. So it is a bit of a trade-off. So we tried to come up with this complexity measure here. So you just take every cell population, you calculate the correlation to every other cell population in the data, and then we take the mean over the different cell types. So the higher this value is, the more complex it is, because that means that the cell types are very correlated. So just take it as 1 is very complex, 0 is not complex. What you see here is that the data sets that are very complex, the most complex one, this is PBMC data. Only 11 populations, but it is very complex. And indeed, if you look at the UMAP of the data, for instance, differentiating things that are supposed to be easy, like CD8s and CD4, is a bit difficult, especially the naive CD8 and naive CD4 are fully overlapping in the transcriptome. So if you only rely on the transcriptome, this is a difficult problem to solve. The Allen mouse brain data at 92 labels is very difficult. Again, this is mainly because it's a very large set of labels, and a lot of them have very few examples to learn from, and so on. Yeah, so it's a bit of a trade-off in between both. When we look at the performance across the data sets, you don't see a clear correlation with tissue, but for instance, again, these are all the pancreatic data sets, so they are certainly easy to do. PBMC remains one of the most difficult ones, I would say, only based on transcriptomics. Okay, so the second set of experiments is a bit more realistic. So we take one data set, we train the classifier, and then we take another data set, same tissue, and then we test the classifier, right? So this is a bit similar to what you would want to do in reality. To try to test this a little bit, there was this paper back then, it was, I think, a preprint, where they tried to compare different protocols on the same sample. So they had cell lines, they had PBMCs, and they had mouse cortex. So we took the PBMCs. This is human PBMC data. They had 2 replicates of the same sample, and then it's tested using different single cell protocols. So we thought, this is interesting. So we can train on one replicate of one technology, test on the second replicate of the same technology, but we can also take replicate 1, train on, I don't know, 10x version 3, test on indrop, and so on. Yeah. So we thought this would be fun to do. So the way I'll show you the results here is that, again, these are the classifiers. I'm showing you the median F1 score, so more yellow is better. This is the data set that we are training on. So in this case, 10x version 3. And the performance here is for these different technologies. So I train on 10x version 3, and I test on SmartSeq2. Yeah. And then the last column over here is that I train on 10x version 3 for replicate 1, and I test on 10x version, I train on version 2 replicate 1, and I test on version 2 as well, but for replicate 2. Yeah, so this should be perfect, let's say. Yeah. You can also do all possible combinations, of course. So you can now train on V3, test on the others, and so on. So this is a big figure, so let's try to zoom in a little bit. We summarize the performance per classifier, so that's what you see here. So these are the classifiers that are doing the best on this data. Again, you see here SVM rejection is not particularly good in this case, so it's not the best performing classifier. If you look in this row, you see that it is actually doing quite okay, except for situations where it has to do with differences between droplet and well-based methods, right? So it's blue at SmartSeq, it's here a bit of blue at CellSeq, and it's always whenever it has to do a combination of droplet and well-based, it doesn't do very well in this case. But still, the normal SVM is doing quite okay. Yeah. You can also summarize it per technology. So let's look at a few things here. So the first one is if you train on 10x V2, test on 10x V3, the performance seems to be particularly high. So the correspondence between the two technologies seems to be quite high. Again, I already said this, SmartSeq2 going from SmartSeq to droplet-based methods is a bit more tricky. So if you train on SmartSeq2 and try to predict droplet-based data, things become a bit more complex. Yeah, one thing we noticed, and it was a bit funny, is that all classifiers are doing particularly worse on the 2 replicates from CellSeq.
Can I ask a question regarding the SmartSeq here? How is it normalized? Is it TPMs or counts or?
Yeah, good point. It is TPM, and then we just do the log normalization. So that can definitely be a problem that you should treat this differently. Our argument was that, you know, if I have an atlas, then I should not have to make these choices. But you're absolutely right.
Yeah, no, but TPM should be the most similar to UMI still, so that makes sense.
So yeah, the observation on cell-seq was a bit surprising, but actually going back to the paper where they generated the data, they also talked a bit about replicability of cell-seq data. So that was a bit funny to see it out of the classifiers. I to be honest, didn't know this beforehand. Okay, so the third experiment, moving to that, is about rejection. So in a lot of situations, you actually want your classifier to reject cells. I think this is one of the most important features of classifiers, that they tell you that I don't know what this cell is, because it's very common that the sample that you're generating is not a replicate of the atlas or the training data. You're generating it for a reason, right? It's either diseased cells or a different species, or there has to be something interesting in it, and that something is often a different or a slightly different state or cell type. The most common experiment to do in this case is that you take, you know, very different data sets. So here we train on PBMC, we test on pancreas. And then ideally, this should all be 100% because the classifier should reject all the cells. Yeah. You see that this is not particularly the case. So a lot of classifiers actually find this difficult to do, even though these are like ridiculously different tissues, right? Here we train on brain, we test on pancreas, and then still this LDA, naive LDA classifier still can classify 95% of the cells and not complain at all. So this is a bit iffy. We thought about a more realistic experiment where you are still within the same tissue, but you have a missing cell population. So here we kind of remove T cells from the training, and then we put them in the testing. And again, the classifier should say, I don't know these T cells, I've never seen them before. But apparently T cells would still be very similar to one of the classes that have been used for training. So the classifiers cannot reject it easily. And then we tried to, you know, remove more fine annotated cells and then see if this improves or not. There's a bit more discussion on this in the paper, so please have a look at it. The only message is that these rejections are really, really hard to do.